Okay, well, welcome to OsteoBites, everybody. My name is Anne Graham. I'm an OsteoWarrior and Executive Director of MIB Agents. Today, we get to talk with 2019 MIB Agents OutSmarting Osteosarcoma Research Award recipient, Dr. Alex Huang. He will be providing an update on myeloid stimulation therapies for osteosarcoma and other sarcomas too. Dr. Huang is a professor of pediatrics and director, Center for Pediatric Immunotherapy at UH Rainbow Babies and Children's Hospital. He is also the co-leader for the Immune Oncology Program at Case Comprehensive Cancer Center. He received his BS in chemistry and MS in biochemistry and molecular biology from the University of Chicago, and then entered the medical science training program at Johns Hopkins University School of Medicine, where he completed his MD and PhD training. Before we get rolling on Dr. Huang's update, I need to explain that I'm not all sparkly today, just because I wanted to look good for OsteoBites, although clearly that is the case as well. But it's because it is Childhood Cancer Awareness Month, and MIB Agents has bows, lots of bows that are being made across this great country by families who have been affected by osteosarcoma. Please go to our website and help spread awareness and sparkles by putting a bow on it. I can tell you that the impact of these bows is significant. It not only helps fund better for childhood cancer, specifically osteosarcoma, but we've had families who have had a kid in treatment and they're really struggling, and for them to come through their neighborhood and have bows on mailboxes supporting them is an astonishingly brilliant and supportive way to help a family through. Also, did I say there are clinic packs for research institutions, labs, hospitals? So you can get a pack of 10 bows and have them on your bows in your hospital or your research lab too. Just go to mibagents.org/bows and make it better with bows, and they're really pretty. Dr. Huang, would you get started with the introductions, please?
Sure, thank you so much. It's been a pleasure to come back again after, I think my last time was in May of 2020. My name is Alex Huang, a pediatric oncologist and hematologist and professor of pediatrics at Case Western Reserve University. Just very happy to share with you some of the updates.
I'm Amy Woodchuck, physician assistant, pediatric oncology, osteosarcoma advocate extraordinaire.
Hi, everyone. I'm Shannon. Very happy to be here today. I am a junior board member of MIB, and I'm also a osteo sibling.
Well, thank you very much, everybody, for this opportunity to update everyone on projects that were catapulted actually by the award that we have received from MIB Agents in OutSmarting Osteosarcoma in 2019. And in the next 40 minutes or so, it will not be a terribly in-depth scientific discussions, but I wanted to bring to everyone's attention 4 projects, 2 of which are pretty close to clinical application and 2 are still under development, but hopefully you will see some translational effort on those fronts as well in the near future. And so I will focus on discussions regarding myeloid stimulation, and this is interesting. For those who are not very familiar with it, it's a type of immune cells found in your body, and that's very prevalent in diseases like osteosarcoma. So I'll get started from there, and hopefully this will always get the dreaded clicking noises. Here we go. So disclaimers, I will be discussing off-label use for natalizumab for the first part of the discussion, and also vectosertib, which is a new agent to target TGF-beta, and then cryoablation will be the end of my discussions. And then there were some financial support over the last few years as a result of some of the pilot studies we're able to gather through funding agencies such as MIB Agents. Just to recall, it seems like so long ago, but it's 2 summers ago that we were fortunate to be visited by MIB agents. These were taken on a very sunny day in Cleveland, unlike today. But look very fondly back on those days, that particular visit, and many things have since then been developed, and I want to update everybody. So the outline of what my brief conversation today will be focused on 4 projects, and I'll spend maybe 5-6 minutes on each, but really to just bring everybody up to date on what's the latest development in each of these areas. So the first 3 projects are what was proposed in OutSmarting Osteosarcoma proposal, including a novel therapeutic approach to actually target metastatic osteosarcoma using an FDA-approved drug, natalizumab, that we term integrin therapy. The second is the update on the development of a novel plant-based water-soluble BG34200 from Oat brand. And then the last part of the first half is an update on our work with Mepecto in South Korea on using orally available TGF-beta inhibitor pills, vectosertib, in osteosarcoma. And then I'll end by discussing some of the novel discoveries of trying to understand why cryoablation, which many of you may have heard as a modality to debulk tumors when they're inoperable, trying to understand why cryoablation sometimes works and sometimes don't work in sarcoma. And hopefully they will provide some future directions. As we all know, in tumor treatments, we have historically focused on tumor intrinsic vulnerabilities, whether it's genetic mutations we can target using drugs specific for them, cell cycle progressions, the basis of which many of our chemotherapeutics are utilized, or protein synthesis and turnover pathways, metabolomes, nutrients, and stress response. So as a result, chemo, radiation therapy, and surgery is the backbone of many of our sarcoma therapies. But more recently, we're also starting to really appreciate the fact that non-cancer cells within the tumor are also very important in providing a nurturing environment for the cancer tumor to grow. And so if we can target some of these factors, such as genetic factors or immune suppression, which is what I'm going to focus today, and even vascular supplies or nervous innervations and microbiome, all play a role, and these opportunities still exist out there for us to actually take advantage of. So I'll focus on the immune aspect of this, and obviously we've been very interested in immunotherapy, and it's not a new concept per se. The concept has been around for over a century, in fact, but more recently with the development of technology and our ability to understand more and more on fundamentally how immune system works, both in cancer as well as in infections and vaccines, we realize that we can take advantage of our very powerful immune system within our body to do some harm against the tumor. And this immunotherapy provides very powerful effects against cancer cells. They can adapt over time. They are durable because of the way immune cells are constructed, and that's how biologically how they function. They can travel throughout your whole body and actually eliminate potentially microscopic tumor cells that you don't see by CT scans and MRIs. They sometimes can synergize with chemotherapy, radiation, and other effects, and it can be highly targeted. The way they recognize their target can be very sensitive, and they can be universal, meaning that if you have different histologies of cancer, if you have similar targets the immune system sees, you can actually apply the same therapeutic modality across histological tissue types in different cancers. And this is why you're seeing different immune checkpoint blockades being FDA approved for bladder cancer, for lung cancer, for all kinds of things, even though it's the same agent across the board, okay? So I want to bring everybody's attention to the tumor microenvironment. We've historically focused on when you take a tumor out under biopsy, you look for tumor cells and see what they look like. But now more and more we appreciate they actually allow more different types of cells besides tumor cells that consist of the entire tumor bulk. And some of these are myeloid cells, which are cells derived from the bone marrow, and they are the myeloid lineage as different than the T cells, which are the lymphoid lineage. And the myeloid cells can be subcategorized into macrophages, dendritic cells, or myeloid-derived suppressor cells. Needless to say, there are different types of immune cells, and the contribution of which in cancer is beginning to be more and more appreciated, and we're starting to find ways to actually manipulate them. This is just a data already now 15 years ago to show that the difference between most adult cancers, here denoted AD, and pediatric cancers across histologic subtypes, by and large, there's a paucity of T cells in pediatric cancer compared to adults. And this is denoted by CD3, which is a T cell marker. And on the other hand, CD68, which is a marker of these myeloid cells that I mentioned earlier, seems to be predominant in many of the pediatric solid tumors. And this is true in some adult tumors, such as pancreatic cancer, for example. And exactly what do these cells do? It's been shown now, Richard Gorlick and others have shown that, especially for osteosarcoma, patients that have a lot of these infiltrating myeloid cells in their osteosarcoma tumor mass tend to have worse outcome compared to patients whose tumor harbors fewer of these myeloid cells. So we know the presence of these myeloid cells have some prognostic effect on the long-term outcome of these tumors, but exactly what do they do is under a lot of investigation. And there's been a very recent development out of pediatric oncology branch from Rosie Kaplan's laboratory, a very nice paper in Cell, that demonstrated in the model of rhabdomyosarcoma that you can actually ex vivo in the petri dish grow up these myeloid cells and genetically manipulate them to secrete factors like IL-12, for example. So you can make almost like a CAR T cell idea. You expand these cells in vitro in a culture dish, genetically manipulate them, and then give it back to a mouse that harbors the rhabdomyosarcoma. And by doing that, you can actually reverse the growth of the tumor, and you can act synergistically, coinciding with chemotherapeutics. So it opens up a whole different arena now besides CAR T and NK cells. Now we can actually start thinking about how do I use myeloid cells that I can grow in culture and give it to patients. So it's a myeloid adoptive cell therapy, and rather than a T cell adoptive therapy. So we are now in a new era, but also demonstrating how important these myeloid cells are. So very quickly, where are we now in all of our efforts to target these myeloid cells? The first is the integrin therapy, which I mentioned several times, including Factor last time. This project is based on the fact that we identify in mouse models of osteosarcoma, the cells that tend to metastasize to the lungs overexpress a molecule called VCAM1 or CD106. And this molecule is absent in primary osteosarcoma tumors that do not metastasize to the lungs. So something about this molecule presence that allows this tumor to grow better in the pulmonary microenvironment. And we suppose that the interacting partner on the macrophage side in the lung is a molecule called VOA4 or alpha-4 beta-1. Okay, full alphabet soup. But suffice to say, why are we interested in VCAM1? The VCAM1 project started when we were looking at cervical cancer and breast cancer, and somebody beat us to the punch that in breast cancer, similar to what we observe in osteosarcoma, the cells actually metastasize from the breast tissue to the lungs, also overexpress VCAM1. And if you knock down VCAM1, you can reverse some of these metastatic potential. So we looked at osteosarcoma in human genome databases that VCAM1 is overexpressed in many of the osteosarcoma samples, although we don't know if most of these are from primary, and we don't know what the source of these VCAM1 expressions are. They're not necessarily only from tumor, because other cells can also express it. But we also looked at pediatric cancer databases from St. Jude database, and we saw that VCAM1 is also expressed at a pretty relatively high level in osteosarcoma and other sarcomas as well. And we took an approach using mouse models of osteo, and the line we use is K7, and there's a metastatic prone subline of K7 called K7M2, where the K7M2 will spontaneously metastasize to the lungs, but K7 is less likely to do so. And we looked at specifically the expression patterns of these CD106 or VCAM. The hypothesis being that K7M2 will express higher VCAM because they metastasize to lungs, and the non-metastatic ones will express less. And lo and behold, that's true. We looked at VCAM expressions by flow cytometry compared to K7M2 as higher. And we actually can genetically knock down this K7M2 cell lines and show that when you genetically remove this VCAM molecule, it no longer can metastasize to the lungs, demonstrating the importance of that molecule. Now, it's hard to go in and try to genetically manipulate every tumor to make sure they don't express VCAM, right? This is kind of hard to do it, across the board. But what we found was that we asked the question whether their expression of VCAM on the tumor cells are truly dependent on the presence of a macrophage in the lungs. So we can do that by using chemicals that deplete macrophages completely from the lungs. And when you do that, you can actually prevent pulmonary metastatic disease of osteo happening in animal models, demonstrating that the presence of the VCAM in tumor and the presence of macrophages in the lungs are the two ingredients that are very important to make sure that these tumors metastasize and take hold in the tumor, in the lung tissue microenvironment. Now, we can't go around and deplete everybody's lung macrophages. I mean, that's important for you to take, to take care of all the infections. So fortunately, we're able to actually identify, and there are in existence already, antibodies that block this interaction between VCAM1 and its partner alpha-4 beta-1, either antibody or specific molecules that block these interactions. And sure enough, if you were to apply using the antibody into an animal that have already established tumor and metastasis in the lungs with osteo, you can actually render these animals long-term tumor-free, suggesting that you don't have to go and genetically deplete every VCAM expression in tumors. You don't have to, deplete in numbers all the macrophages, but you can actually functionally block these interactions using antibodies targeting VCAM, and you achieve the same results. And there's just a couple of examples, functionally how exactly what happens when you do this blocking experiments. What we show is if you block these interactions, you enhance the NK cell activity, you change the morphology of the macrophages to be more pro-inflammatory or anti-tumor. So the whole environment is now changing because of this one manipulation you did to the tumor. You reverse the tumor from being very prolific now to regress. And more recently, we have now demonstrated that this interaction is very important for the production of something called arginase 1. It's an enzyme that is very abundant in macrophages that are more immune suppressive. So, by taking away this VCAM, you actually reduce the immune suppressive factors that set up the entire microenvironment to allow the tumor to thrive. That, at least that's what we think is going on. Now, based on that, we were able to compose a phase 1, phase 2 study, single arm open label proof of concept using FDA approved antibody drug that targets this alpha-4 beta-1. And this is already FDA approved for multiple sclerosis and inflammatory bowel disease, made by Biogen. And Kristen, who's been on this case for 3 years, God bless her. We went now as far last time when we spoke to you, we were just executed a contract with a company. They agreed to sign on after 2 years of talking to them. They agreed for us to file an IND with the FDA last May. And I just here to report in December of 2020, we got an IND approved from the FDA. And subsequently, we now have funding for the phase 1 portion of this trial. I mean, it's still in the process of funding the phase 2 trial. Anybody with extra cash, I will give you my my contact information, and I'm all your friend. But we now finally, after another 3 or 4 months of going back and forth, the final IRB approval is actually that meeting right now as we speak. So right now, September 9th. So hopefully by tomorrow morning, I'll hear anything. Once it's open, we have initial training for our staff, and the drug is already in our pharmacies, ready to go. So this trial is targeting for unresectable pulmonary metastatic osteosarcoma, okay? It does not address in primary lesions or elsewhere multifocal non-pulmonary osteos, because for whatever reason, it's still unclear to us. This VCAM1 VLA4 interactions is very specific for pulmonary, maybe bone marrow, but pulmonary microenvironment, but not so much in peripheral muscle and other tissues. So it's a very tissue-specific interaction, and that's why our protocol does not address non-pulmonary recurrent or metastatic osteosarcoma for this clinical trial. Okay, so that's where we are. That's the first one that hopefully will be open very soon for unresectable pulmonary metastatic osteosarcoma. The second update is BG34, which I don't know if you remember my discussion from last time. It's a molecule that is derived from the outer shell of brand. And this is a brainchild of Dr. Mei Zhang, a very brilliant assistant professor in biomedical engineering and now at Case Comprehensive Cancer Center at Case Western Reserve University, who isolated this from the outer shell of oat bran, parts that we throw away when we eat oatmeal. But it turns out that there is a component of the outer shell that has the beta-glucan structure of 1,3,1,4 sugar linkages. Is a linear structure that you can isolate very high purity, and it's water soluble. It's almost like a soluble fiber that you hear about. But what's interesting about this molecule is that you can isolate them at different molecular weight. And there's a specific molecular weight, BG34 at 200 kilodalton. This hence the name BG34-200. That has a very specific immune activation function that its partners BG34 10 kilodalton or 500 kilodaltons do not have. And this is a conundrum we haven't completely figured it out yet, but there are many aspects of this mechanism we are now starting to understand further. This is just to show you on the left here is a mouse model of a melanoma, where if you treat mouse-bearing B16 melanoma with BG34-200 in this very specific range, you have shrinkage of the tumor by just single injection alone. And whereas 510 do not achieve the same thing, and the controls are either dextrans or PBS. And we have done so similar trials in using the earlier K7M2 pulmonary metastatic osteosarcoma, and show that BG34-200 also can actually rescue animals that develop pulmonary metastatic disease. And we came in 10 days, 14 days later with this BG34, we're able to render a majority of these animals tumor-free, at least by detection using luciferase imaging. And it's very exciting development. Because of these data, we actually may have gone and we received both an orphan drug designation and a rare disease designation from the FDA in August of last year for the treatment of osteosarcoma. And just most recently, she has received a DoD award, $1.6 million, to study this further in sarcomas and other cancers. And then we have most recently received a Coulter Award for the development of this drug in the GMP production. By the way, we're doing this in our lab. We don't have a factory to make this. You've got to go buy kilograms of bushels of wheat and isolate this thing and go through a whole product characterization with the FDA. But this BG34, we now have shown that it actually enters into these myeloid cells via the surface molecule CD11B, and it induces the maturations of these myeloid cells through a mechanism we're still studying, and also induces the development of CD11C positive dendritic cells, making the whole tumor microenvironment from a cold tumor microenvironment to a hot tumor microenvironment, increase the number of T cells infiltration, increasing the function of these myeloid cells from an immune suppressive to an immune stimulating phenotype. It's a very interesting molecule, and just stay focused, stay tuned, because I think more will come from here. But before we get to all the molecular details of how it works, we couldn't wait to translate. So we have already been in discussion with Amy LeBlanc, the NCI, with the with comparative oncology branch, to think about how do we translate this into patients ultimately. And we decided that the first cohort of patients we're going to try with the BG34 will be canine osteosarcoma patients. And so this actually it's already been drafted. And we're just waiting for the production that we will give to our canine friends. and then also funding to actually run the first clinical trial in canine. So keep an eye on this. This is going to be hopefully imminently for the canine osteosarcoma for BG34. And obviously the hope is that we'll then push forward with the data we obtain from the canine to provide both the safety data as well as some toxicology and PK/PD studies that will then ultimately go into a final IND submission for human trial, hopefully in 2022 or 2023. And then the third project I'll very quickly just summarize is our work on vectosertib. And this is to target another very immune suppressive molecule in the tumor microenvironment called TGF-beta. And this is something that many people know is a very important molecule to target. And historically, it's been very difficult to target. But this is all work done by Sung-hee in the laboratory in conjunction with our very good friend. And we're very fortunate to have this partnership with Mepecto, the CEO of whom is Sun-jin Kim, who used to be at the NCI. And now they're based in South Korea. They have developed a orally available small molecule inhibitor against the receptor for TGF-beta receptor type 1. And this is the molecule that is highly potent. So this is showing here is what happens if you take this pill by mouth. And within 2 hours, you completely shut down TGF-beta signaling in your peripheral blood. And that effect, once you stop the drug, that effect actually can reverse back to normal within 24-48 hours. So it's a drug that you can take in, within an hour, 2 hours to 4 hours, the drug has already taken effect. And if there's any toxicity, you can stop within a day or two, the drug will disappear. And so it's a much better, well-controlled drug than an antibody-based drug, for example, which once it goes in, sometimes it stays on in your system for a whole month. And this is very potent in terms of inhibiting TGF-beta signature here, shown here is something, SMAD2 is something that we look for as an indicator of how well it works. And this cuts across both human osteosarcoma cell lines as well as mouse osteosarcoma cell lines. Furthermore, you can treat these the cell lines of osteosarcoma and inhibit directly the tumor growth. So this drug has an effect directly on tumor itself. But we also know the drug also has effect on the immune system that surrounds the tumor cells. So in both cases, it's an enhancement of the immune system and suppression of tumor growth. So it actually is a two-pronged approach to actually killing the tumors, having the same target, but different populations within the cell. So you can see the tumor growth potential decreases as you increase the dose to be somewhere between 1 and 2 micromolar of this drug. You can shut down most of the osteosarcoma in human. It's interesting that 143B, which is c-MYC amplified, does not. And you can see that it shuts down SMAD phosphorylation in 143B, even though the MYC does not get shut down. But interestingly, in other cell lines that we tested, this TGF-beta inhibition can abrogate the expressions of MYC in many of the osteosarcoma cell lines we have tested. And so and also, as far as signaling cascade is concerned, we know that we are perturbing some of the pro-growth signaling cascade in the osteosarcoma specifically. And this is something we're still working on, exactly what happens from a molecular mechanistic standpoint. But in terms of applying in vivo, we've done multiple experiments now. This is done using human osteosarcoma cell lines, SLS2, in immune deficient animals. And then we feed this animal by gavage, this vectosertib pills. And we can actually substantially reduce the tumor incidence in many of these mice downward from 100% of tumor incidence down to 40%, meaning that 60% of the tumor will not develop osteosarcoma, even in an immune deficient animal. So showing that there's a direct effect of the drug on the tumor itself. What's actually very exciting is the animals that actually over time, in the beginning, we treated for 30 days and there's no evidence of disease. And by 60 days, the remaining tumors, they grew out. Okay, they have been exposed to the drug. They now they relapsed, they grew. If we were to treat these animals again with the oral vectosertib, you can actually sustain the suppression of these tumors for prolonged periods of time, and which is a very, very exciting development for us. And so, and this also can be shown using going back to mouse models where there are immune system involved, as well as direct toxicity of the drug against the tumor itself. We see again a very potent inhibition of the osteosarcoma growth. We see changes in the immune microenvironment of these tumors. Again, we see increase in activity of NK cells, these anti-tumor macrophages, increasing in T cells. And what's even more exciting is that if you combine this TEW or this vectosertib drug, the oral pill, in conjunction with PD-L1 antibody, which historically does not work very well for osteos, everybody knows, we now have this synergistic effect, suggesting that maybe this vectosertib, not only is it targeting cancer cells directly, it's activating immune cells such that now they can respond to anti-PD-L1 antibody. And based on these data, the pharma company has just filed a orphan drug designation with the FDA. And as of two Mondays ago, the press release came out, it got granted the ODD designation. Very excited about this. And the company's now working very hard with Kristen in the lab to actually draft a concept for a clinical trial based on what we've shown them. And I'm just, I'll take a little bit of credit to be the one that pushes the company to think about osteo, because they were not thinking about any AYA sarcomas for sure when we first talking about this. So this is where we are in the initial 3 projects that was part of the OutSmarting Osteosarcoma since 2019. For the first integrin therapy, as I said, hopefully we'll find out from IRB meetings today. We'll see where the status of that is. The BG34, we have already a pre-IND meeting with the FDA in 2020, sorry, this is 0 here. As I said, there's a canine trial that's pending. We received the RDD and ODD status from the FDA back in August last year. And a new award's been awarded from the Department of Defense to Dr. Mei Zhang. And then for vectosertib, a ODD designation in August, just 2 weeks ago. And then the trial concept is being drafted with Mepecto as we speak right now. In the last few minutes, I know I'm supposed to finish in 5 minutes, 10 minutes. Because if you don't stop me, I'm just going to keep talking. There's a lot of things to talk about. So I want to maybe just the newest development in the laboratory, and this is actually a work that's been ongoing for 3 years now, 3 or more years, about cryoablation in sarcomas. And I think the concept may not be so foreign to many of us on this call. And cryoablation is basically a clinical application where you pump ultra cold liquid nitrogen or argon gas through a probe that the radiologist can see under guidance, either ultrasound or CT guidance. And the concept here is that in inoperable tumor or sometimes a nerve bundle, you really need to sort of ablate it because it's causing so much pain. You can pump liquid nitrogen into the area, and it does several things. One is the ice crystal will clog off the blood vessel. So you basically strangle the tumors to death. And the second is you freeze the tumor cells, and then the water crystal then ruptures the cell membrane, and the cell died of these necrotic cell death. Now, the necrotic cell death is actually a form of very inflammatory cell death that has a role in immune cell activation. And as a result, there's been many conversations whether cryoablation may be a way to actually as an adjuvant to immunotherapy. And can you take advantage of the fact that many of our sarcoma patients might actually go through these kind of procedures? Is there a way to understand, how can we take advantage of this procedure in terms of immunotherapeutic development? And we are not the first one by far to think about this, including Jim Allison's lab, a Nobel laureate, who has actually shown that you can combine anti-CTLA-4 with cryoablation and lead to synergistic effect immune activation in some tumor models. And again, the idea here is that cryoablation induces necrotic cell death by rupturing the cell membrane, and it elaborates a lot of contents from the tumor cells, some of which are very immune stimulating. And that is part of the overall efficacy. And once you stimulate the T cells or immune cells in that tumor where you cryoablate, these cells can then travel throughout the whole body and then kill other non-cryoablated tumors. So this is something we call a scopal effect, right? So you put your probe in one site, and if you have 2 or 3 other lesions in the other lungs, people have seen regression of the tumor in the other lungs where you did not treat. This is something we call a scopal effect. And we think that's immune mediated, and I'll show you some evidence of that. Now, the problem is for many clinicians on this call, you'll know as soon as I talk about cryoablation, people say that doesn't work. Because invariably it comes back, and it comes back faster than usual, and it's just inconsistent. And sometimes I've spoken to quite a few people that are doing this, and there's paradoxical outcome. Sometimes the doctor might go in with trying to cryoablate, and they are very thorough. They're trying to ablate all the tumors, and the tumor comes back faster than before. And sometimes you see better results when you partially ablate the tumor and leave some live tumors around, and the tumor actually shrink. It's a very, confusing picture, and we want to know a little bit why that is. One of the pathway we've focused on is this pathway called STING. It's a very ancient myeloid specific detection of bacteria and viruses. It's a way for the myeloid cell, when they are infected with DNA viruses and other things, the presence of naked DNA, which should be in nucleus. But if they ever happen in the cytoplasm, it's an activating signal for this pathway called STING. And it binds to dinucleotides or two DNA sequences that translocates to the Golgi from ER, activates a whole transcriptional activation pathway, and makes cytokines that attract immune cells to come and destroy this potentially virally infected cell. And so the importance of STING pathway has been very well described in myeloid cells. And we wanted to know whether it's operational in tumor, whether the STING pathway is what's responsible for the responsiveness or unresponsiveness of cryoablation in tumors. In this case, sorry, we were using rhabdomyosarcoma and not osteo yet. We will be there, we're not there yet. But using mouse models of rhabdomyosarcoma, Mohammed has shown in the lab that if you co-culture these cells undergoing a cryoablation with bone marrow derived macrophages and dendritic cells, you can lead to the immune activation of these myeloid cells. The idea being that when you cryoablate cancer cells, it will rupture the membrane of the cancer cells, and the contents, including double-stranded DNA, will be leaked out into the media, and that DNA can be picked up by the macrophages that activate the STING pathway, leading to immune activation. Okay, that's the hypothesis. And so, sure enough, we showed in animals, we subject the animal to injections of a tumor, wait till the tumor becomes very big. And this is a very aggressive rhabdomyosarcoma cell line. And we can cryoablate these tumors by touching the skin with this very cold liquid nitrogen. And sure enough, you can actually reduce the tumors to no growth at all. And the myeloid cells in that tumor microenvironment are all immune activated. The metastasis is decreased. And so this is all good. Now, the question is, in this model, the tumor did not grow after cryo. Is that because we freeze off the tumor? They're all dead, right? That's why they didn't grow. But what Mohammed showed was, in order for you to have that sustained suppression of tumor, you actually require the presence of immune cells. Meaning you do the same thing in the immune deficient mice, the tumor grow right back. Even though you freeze the tumor to death, you think, but there's enough live tumors left to come back and overtake this host, and eventually will kill the animal. And same thing, in an animal that responded to cryoablation, if you remove either CD4 T cells or CD8 T cells, you lose this therapeutic effect. So you need the immune system to be intact for this to work. And there's evidence that after cryoablation, the immune activation status is improved. I'll skip this page, but the bottom line here, what we found was that, very interestingly, the importance of STING is not only solely attributable to the myeloid component. what Mohammed was able to show is that tumor cells themselves, if the tumor cells also have intact sting, live tumor cells will respond to the same thing as the myeloid cell, and both the myeloid cell and live tumor cell will contribute to this immune activation. And the way we show that is if you were to genetically knock out sting either in the immune cells or in the tumor cells, you lose this protection, okay? You lose the protection of cryoablation in terms of tumor growth, and you really need the presence of sting both in the tumor and in the myeloid cells for the cryoablation to work. Now, historically, what we are focused on is sting activation in the myeloid cells. And this is, you might have heard sting agonist therapy, intratumoral sting agonist therapy, and pharma companies all focused on that. But what we've shown is that tumors expression of sting is actually a major contributor. And I'll just skip this. What is interesting is that many of the tumors we looked at is very heterogeneous when it comes to sting. Many tumors have turned off the sting pathway. And that is perhaps attributable or contributes to the reason why some tumors do not respond to cryoablation, because epigenetically or genetically, the tumor cells turn off the sting, thereby making it a very cold microenvironment, despite the myeloid cell trying to activate its sting and trying to keep a warm, tumor microenvironment going. And this is sort of some of the evidence comes when you see, as you look at databases of sarcoma patients, as you go through the initial diagnosis to metastatic disease to relapse, there seems to be a trend of decreasing sting activation as you go through different stages of disease development. And there's some evidence that depending on the histological subtypes and mutations contained within, the sting pathway is different. And we don't have all the answers to that yet, but we're now teaming up with MD Anderson to look at a lot of patients who have gone through cryoablations to look at their initial tumor biopsy, how many patients have high sting expression, how many patients have low sting expression, and based on that stratification, what is the clinical outcome of these patients undergoing cryoablation? Is there a direct correlation between high sting, better outcome with cryo, low sting, worse outcome with cryo? And if so, then the next question is, can we somehow use drugs that we already have on the shelf to actually increase some of these pathways that sting has, or the sting related pathways, to make these tumors more responsive to cryoablation? So I'll end there exactly at 39 minutes. But here is the overall summary. So I've talked to you about the first 3 projects, and the newer project, it's there's a newer clinical concept, and I need to talk to a few important players, including the company that is very generously supply us with the cryoablation machine. What would be a clinical trial concept that we can emerge from this discovery, and whether we can correlate the presence of sting and the degree of sting activation in sarcomas, and then the clinical responsiveness to cryoablation, and then ultimately what turns sting off in osteo, what turns sting on, how can you turn it back on, and which part of the sting pathway, signaling pathway, is important. I mean, it's a sequence, this whole slew of molecules. Is sting itself the important thing, or the subsequent players downstream of that signaling molecule more important? We don't have all the answers yet, but this is just another area of investigation going forward. And the last slide before the acknowledgement is what's on the horizon. So as you see, we have several projects going on, brewing all at different stages of translation, some in early discovery, some translational, some ready for clinical translation. And to coordinate all that, one of my newer projects here locally is to establish a Center for Immunotherapy based at Rainbow. And the idea was to be actually a hub to take into all the discovery science that's happening on campus and elsewhere, to partner with industry partners and take advantage of some of our production, cell production facilities and others, to really address using immunotherapeutics to address pediatric and AYA cancers, but also with the advent of immunotherapy in other arenas to see whether or not we can actually help other children with different diseases that could be helped with immunotherapeutic approaches, including GVHD, including inflammatory bowel disease, including allergy infection, and one of my pet peeves actually, even for diseases like autoimmune autism, some of the restoring some immune balance to the host, there's some suggestion that it might help the overall severity of autistic spectrum in some animal models, which is actually very interesting, but we're not there. So here's the acknowledgement. I don't do any of the work. I'm a mouthpiece for everybody. Here's a more recent picture, including some of our summer students. Many of the folks whose work I mentioned, Sang-hee was on the TGF-beta experiment, and Suzanne did a lot of work on looking at STING genetics, and Muhammad is working on, he's the one that give credit to him for thinking about the tumor STING status versus what everybody else is thinking about myeloid STING status. Always glad to hear students who think against the grain and come up with new ideas. And Muta, Melissa, and Ali, they're all MD PhD students working on other exciting projects that hopefully we'll have some time to talk about in the future. And Kristen's been a driver in translational clinical trial drafting, and Yami's working on her way in different in leukemia models, which I don't have time to talk about in this audience. But Jay and Sada makes everything works, and Miranda just joined us recently in applying to graduate school. So obviously, thank you for trying to make it better, and we are all on the same team, and this would not have been possible without the support of folks on this call, and hopefully we'll have more news to come in the very near future. So thank you for this time, and gosh, I went over 5 minutes.
Yes, thanks. We expect it. But it's totally fine, because all that information was just spectacular and so hopeful and interesting. And we also, it's not like a conference where I'm like, okay, we got 20, you got 20 minutes and we get the hook out. So happily OsteoBites, we get a little bit more leeway. Okay, go ahead, team, what you got?
So I have a first question, which is, in the anticipated trial with integrin, could you give the parameters for patient participation?
Yeah, so again, the basic thing is that the patient will have to be, it's from age 5 to 30. So it's a wide range. We want to capture as many patients as possible. The only caveat here is that this is intended for patients who have unresectable pulmonary osteosarcoma. So if you have pulmonary osteosarcoma plus some other disease elsewhere, you will not be eligible. And the reason is, we wanted to, based on the evidence we have, we've seen that it does not respond to primary lesions, at least in our animal models. Now we might be surprised in vivo, but because the integrin therapy itself is not completely without side effects, right? So patients who get this, especially those that actually got it for prolonged periods of time, for MS therapeutics, there is a risk for severe immune suppression to the point of reactivating some of the viruses that live in your brain. You can get, sort of progressive multifocal leukoencephalopathy that can be lethal. And we don't want, and those are typically patients that have received this antibody for more than 2 years. And so as it stands right now, the trials for patients that fit these disease criteria, and it's up to 2 years. We don't go beyond 2 years. But really the idea is to see, for the first time, demonstrating the same FDA drug is safe to use in cancer patients. And ultimately the goal here, I think, will be to actually develop inhaled nebulized formulation so we can actually target only the pulmonary tree and lessen the potential side effects on systemic circulation. But I can't go there until I engage the company with their known drug preparations. Hopefully we'll get them excited about the initial studies, and then we can talk some more. But this approach, I'll just tell you, is not specific for osteo. We now see the same thing in T-cell leukemias that failed bone marrow transplant. These stem cells upregulate VCAM1, and the same therapy can be efficacious. So stay tuned.
So along those lines, somebody out in the audience is wondering if that integrin trial is possibly available as compassionate use, like if they do not qualify for the study.
Yeah, I think we in fact, we have been in conversation with several families about that. And I think we have approached the Biogen. They have expressed their willingness to help with anything. So, if there are patients that I'm happy to talk to the primary oncologist, and obviously we will respect the primary team to know what's the best scenario for patient, whether this makes sense or not. And in some of our more recent conversations with different families, for compassionate use in other drugs like bactosertib, in those conversations, they have diseases elsewhere outside of the lungs, and for that reason, we direct them to this TGF-beta inhibitor rather than the natalizumab. But the company, when we approached them about a year ago, because they were potential candidates, they did express their willingness to actually work with the physician to file the compassionate use application with the FDA.
Along those same lines for compassionate use, there's a question if your clinical thoughts about cryoablation might be also available with the immune modulators for compassionate use.
Yeah, it's one of those things. I obviously I don't do cryoablation. So I number one, it depends on the clinical scenario, whether or not the approach is safe to do so. And really a lot of it has to do with comfort level of the physician, the treating physicians and their institutions, their experience with cryoablation. Not every institution has a lot of experience with that. And at least in my conversation with our own people here, you get a lot of sort of eye rolling. I was like, oh, I'm not going there. But so it is a bit, I think early at this point to know how generalizable and how aggressive we should push for people to think about this. There are experiences and folks who have done this for a long time, but usually what they do is not a single cryoablation. They couple that with a whole bunch of immune stimulating therapies, most of them is intratumoral. So you cryoablate and then you inject either CTLA-4, toll-like receptor agonist, a lot of sort of soup du jour, trying to activate the immune system. And part of the ongoing debate is because everybody sort of do things haphazardly and randomly and not in a very well controlled way. You never know how to interpret these data. And so this is why part of the driver behind thinking about doing more formalized clinical trial in sarcomas, correlating cryoablation potentially plus minus something else. I don't know what that something else is. And then correlating them with the underlying genetics of the tumor on biopsy. And can we make this tumor sting as a biomarker of response potentially? We don't have that yet. And this is why we're partnering with MD Anderson. Hopefully there will be going through, who are the suitable patients we should look at and look at their tissue microarrays to see, can we draw some conclusions from these retrospective studies before we think about a prospective clinical trial.
Thank you.
That certainly it's a conversation. It certainly is a conversation. I'll just say, from compassionate basis, it's always a conversation you should have with your doctor.
Right.
And so and hopefully people at least will be willing to sort of look through the evidence and contemplate whether or not it's approachable thing, and number two, whether they have the right people in their local institution to do such a thing.
Yeah, thank you.
So I have one last question. What inspired you to become a researcher?
Because that's the only thing I know how to do. No, I'm just kidding. So for several things, I've always been interested in research, and I've shared with this audience before the impetus for osteo specifically is because of my cousin, who passed away from osteo 40 years ago. And by the way, I know my cousin's brother is on this call. I just see his name. Hey, Wayne. So Wayne's brother passed away, and that's been for a long. He just said hi. 40 years, and 40 years later, we still treat kids with the same drug that his brother went through. And so that's really the impetus to really trying to make an impact, trying to do something different. And now, and the other thing is I'm a little ADHD, as you can see. My mind kind of go all over the place. I'm not happy with just doing one thing, and research allows me to kind of just, especially with a whole bunch of very fun people in the laboratory. It allows us to explore things and hopefully in that process bring new things to come along.
I'm just reading Dr. Healy's response here.
The freezing explanation is overly simplistic. I agree with you. And actually, this is actually one of the things we are trying to, and part of the problem also, when you apply these clinically, everybody does things very differently. And some people, swear that you got to do 8 minutes freezing with 15 minutes thaw plus 3 cycles, and just it's all over the map. So exactly how do you control this in a controlled fashion? That's part of the problem. We haven't gotten there, and we just can't. So part of a conversation, ongoing conversation. What would a clinical trial look like? How do you technically regulate that? To a point where you can actually. It's not like we're giving the same dose of drug, right? How do you apply this? I agree with Dr. Healy.
Okay, so just to respond to another question there, it was what was Dr. Healy's question? Dr. Healy said technical issues with cryo that works by other mechanisms than the dogma, i.e. kills by Donan effects, rate of freeze and rate of thaw, number of freezes, etc. The ice crystal explanation is overly simplistic, great potential, and all has to do with eutectic point of water. Did I say that right?
Yes.
I'm always, we are open to, want to converse with people that have obviously a lot more thought into this. Is something that we start thinking about very recently. So always welcome any conversations and discussions. And if you have any ideas, I'm all ears.
Love sharing ideas. I wanted to say to Wayne, I don't know if you shared with him, Alex, that at every Factor conference, we have an angel wall where we honor the kids who have passed from osteosarcoma, and your brother's on that wall. And I have your email, Wayne, so I'll send you a link to that video. It's on our YouTube channel with all of our videos, but I'll send you that specific link. And then we're running out of time, but I wanted to say 2 things in addition to what I'm about to say. The first is I wanted to say Mei Zhang, Dr. Mei Zhang in your lab is not only brilliant, but she is very generous with her time. She helped us create the Chinese version of osteosarcoma, From Our Families to Yours, which is now distributed throughout China. When you get osteosarcoma and you live in China, you get the Chinese edition of Osteosarcoma, From Our Families to Yours, with thanks to Sunflower Children and Shiyu Children's Foundation. So thanks to Dr. Zhang for that. The other thing to say is, if this highlights nothing and everything went over your head, it's okay. What your takeaway should seriously be from a conversation like this is funding research matters. It doesn't happen from the government necessarily or in a big way. It happens regularly with grants from organizations like MIB agents. And the other thing to say about research coming from organizations like MIB is it allows for creativity and, I'm going to say, magic to happen, because you're not so regulated as you are with a grant from the NIH. So it's really important. Again, like you can help fund research with bows, or you can just send us a big check and we'll take care of making sure that it gets to the right person. Also needed to say thank you, Dr. Huang, for all of your hard work. You have lots of messages. I hope you can see them on here from your peers and from patient families who are saying thank you so much for this hard work. And we're really glad that you were, well, we don't love the reason that you were inspired to be a researcher for osteosarcoma, but we're very grateful. And Wayne, your brother's life had meaning because of what Dr. Huang does today for all of us. Thank you to your whole family.
Thank you.
Okay, so gotta wrap it up and give everybody the rest of their day back. Wanted to also add that more information on this OsteoBites and all OsteoBites can be found on YouTube. It can be found on mibagents.org or wherever you get your podcast. If you registered for this OsteoBite session, you're going to be emailed the links shortly. And then also next week, we have another really excellent OsteoBites with MIB Agents 2020 Outsmarting Research Award winner, Dr. Andy Livingston and Dr. Nancy Gordon from MD Anderson. They're going to be talking about hydroxychloroquine and self-eating in osteosarcoma. So bring your snack. Until then, thanks again to Dr. Huang and his team for using their powers for good for OsteoWarriors. And thank you to junior board member, also incredibly brilliant and wonderful and all the good things, Shannon McCormick, and to MIB superhero volunteer and all things wonderful, Amy Woodcheck. And we'll see you next week. Thanks for being here, everybody. mibagents.org/bows. Get your bow, get your sparkles on. Okay, thanks all.
Thank you so much.